We said here that DNA is typically double-stranded inside of a cell, and what holds the two strands together is hydrogen bonding between the nitrogenous bases. It turns out that only certain nitrogenous bases can hydrogen bond with other nitrogenous bases. So here's what we mean, and you might remember this from towards the beginning of the quarter. So base pairing is specific, is what we're trying to say. And what we mean by that is if you consider an adenine nitrogenous base, it can only form hydrogen bonds, it turns out, with thymines. And vice versa, T's can only form hydrogen bonds with A's. If you consider a guanine, it can only form hydrogen bonds with C's, and vice versa. C can only hydrogen bond with G. There seems to be some, or there is some experimental evidence to back this up. So basically the reason why A's can only bind to T's and G's can only bind to C's is because the hydrogen bonding that occurs between the two can only occur if an A is with a T and a G is with a C. So we'll show you a little bit more about that later. But there's evidence to back up the fact that A's can only bind to T's and G's can only bind to C's. And here's the evidence. So people have measured actually the width of a DNA molecule. So there's ways to do that. So people know how wide a DNA molecule typically is. And people have also measured how wide an A is, how wide a thymine molecule is, how wide a guanine molecule, and how wide a cytosine molecule is. And so it turns out that if A's bound to G's, that width, that combined width of the A and the G would be greater than the known width of DNA. If T's bound to C's, the width of the T and the C would be actually more narrow than the known width of DNA. Right? So that's one of the reasons why people know that A's don't bind to G's and T's don't bind to C's. They don't match the known width of a DNA molecule. By the way, if you consider, so we said that RNA has U's instead of T's. So this is just showing that this hydrogen bonding cannot occur. We know that this does not occur. We mentioned that RNA has U's instead of T's. And so you might remember then that what U's are complementary to. So U's have a structure that's very similar to thymine, which means that U's, just like thymine, can form hydrogen bonds with adenine. Right? So if you are looking at a U in an RNA molecule, what it forms hydrogen bonds with, what it's complementary to, is an adenine. So the base pairing is specific. Another thing that's worth knowing about a DNA molecule in terms of its structure is that in order for one strand to be able to bind to its complementary strand, the complementary strand has to be anti-parallel. In other words, it has to kind of run in an opposite direction. So if here's one strand of nucleic acid and it's going to bind to its complementary strand has to be kind of flipped in relation to it. And that's what will allow the hydrogen bonds to occur between the nitrogenous bases like this. And so to kind of show a little bit better why this has to be the case, so why do the two DNA strands have to be anti-parallel in order for this hydrogen bonding to occur? How about if we show kind of a different picture of DNA, one that'll kind of illustrate this better. So again, in order for one strand of DNA to bind to its complement, they have to be anti-parallel. So they look like they're running in opposite directions. Another way of considering anti-parallel is if you look at this strand, the top of it contains the phosphate, whereas on its complementary strand, the top of it is the sugar, right? Its phosphate is down here. So they have to be they have to be running in opposite directions in order to have these hydrogen bonds form. So to see how that is, how about if we kind of enlarge this region here? And you might remember that hydrogen bonds form as a result of oppositely charged or regions of the molecule that have partial charges that are opposite in nature. So you know that if oxygen, for example, is sharing electrons with the carbon, it's more electronegative than the carbon, so it's going to have a partial negative charge. Turns out that nitrogens here are more electronegative than hydrogens, so when these two are sharing electrons, they're going to spend more of their time around the nitrogen, so the nitrogen's going to have a partial negative charge, and the hydrogen's going to have a partial positive charge. And so the hydrogen bonding then between two nitrogenous bases, like this adenine and thymine, specifically occurs between this oxygen, this hydrogen, that hydrogen, and that nitrogen. And then you can see where the specific hydrogen bonding occurs down here between the G and the C. Notice how, again, the two strands, in order for these hydrogen bonds to occur, have to be anti-parallel. So look at the way that the sugar phosphate backbone is going. The phosphate is at the top of the strand. Over here, the phosphate is down here at the bottom. So again, they're flipped in relation to one another. What if you unflip them? Right, so to see what that looks like, we'll enlarge this portion here and show you what it looks like. So what if we took this and flipped it around so that it's now not anti-parallel but parallel to this other strand? So now take a look at the sugar phosphate backbone. The phosphate is up top. Over here the phosphate is also up top. So now they're facing the same direction. It turns out that if they're facing the same direction, hydrogen bonds will not be able to form between these complementary strands. And here's why. So if we kind of draw in where the partial charges would be on this nitrogenous base and this one also, where there's oxygens, there's a partial negative. Where there's nitrogens, there's also a partial negative. Where there's hydrogens, that's where there's the partial positive. So you can see here that they're not lined up correctly in order for hydrogen bonds to occur. Right? You know that a hydrogen bond's not going to happen between two like charges. And same thing here, partial positive, those are the same. So you know that a hydrogen bond will not form there either. So the bottom line is that the reason why, so we'll write it down here. So if two DNA strands are parallel instead of anti-parallel, then the complementary nucleotides, they won't be able to hydrogen bond with one another, like we just showed you here. All right. So again, that's the reason why DNA is always shown like this, like the two strands are flipped in relation to one another. It's so that these hydrogen bonds can form. And again, another way of thinking of anti-parallel is like this. So if here's one strand of DNA, the part that has the phosphate is considered the 5' end. The part with the sugar and not the phosphate is called the 3' end. So if it's going to bind to something that's anti-parallel to it, that means that its complement is going to be flipped in relation to it. So if the top of this one is the 5' end, that means that its matching end would be the opposite, the 3. Same thing here, if the bottom of this strand is the 3' end, then the matching end on its opposite would be the 5. So again, anti-parallel, that's all that means. So one last thing to mention about the basic structure of DNA is that if you take a look at DNA, real DNA inside of a cell, it doesn't look like a ladder like this. So basically it looks like a ladder, but it turns out that it's twisted. So if you take the top portion and then the bottom portion and then twist them, you'll see something that looks like this, a helix. And so in real life, DNA looks more like this instead of this. So it's a twisted ladder is what DNA looks like. So now that we've kind of reviewed the basic structure of DNA, we can kind of figure out how does DNA end up replicating itself. And so here's the basic idea. So if here's a schematic of a DNA molecule and it's color-coded, so here's what a single nucleotide again looks like. It's color-coded so that the green is the nitrogenous base, the blue is the sugar, the yellow is the phosphate. Basically what occurs in order to replicate a piece of DNA is that first it has to get separated. Right? So basically the information on a DNA molecule is locked up in the middle in these nitrogenous bases. So to be able to read that information, you have to get to it. The cell gets to it by separating the two strands.